Hello, this is Mr. Russell, your AP Biology teacher, and today we're going to be talking about eukaryotic cell components. When we look at these, there are three main parts of a eukaryotic cell. Here we're going to look at a plant cell and an animal cell. There's your animal cell. So we look at this, we've got three main parts. You've got the plasma or cell membrane. So in this case you can see it surrounding your animal cell there outside. You've got the nucleus, which is going to be right there in the center. It's going to be the control. It's going to control all the activities by holding the DNA. DNA being the instructions for making, carrying out functions of the cell. Then you got the cytoplasm, or the cytosol. It just creates workroom or space for holding the organelles. I compare this often to a Jello cup of fruit. So if you think about the Jello being the cytoplasm or the cytosol, and then the organelles being the fruit, it's going to hold the organelles in place. If you go back and you look at the nucleus, and we look at it here being right there in the center of the big grape colored round object that has a fourth of it cut out, you can see here the nucleus is a membrane bound structure. It acts as a control center for all activities performed by the cell. It's kind of like the principal's office here at the school. So it is the source of genetic information, or DNA. It acts like a vault for the million-dollar blueprint of the cell. It also has a nuclear envelope that surrounds it that acts as the actual vault itself to protect the nucleus. This is made up mainly of a bilayer of phospholipids. It also contains pores or tunnels composed of proteins for charged molecules to travel through, such as nucleotides to be able to make RNA. These nucleotides come from food. So the messenger RNA, or the mRNA, leaves to help make proteins in the ribosomes, or the construction site, or the cytoplasmic sites. So when we look at this, you've got DNA. This is the million-dollar blueprint. Made up in, you're going to see this in two different phases: the chromatin phase and the chromosome phase. So if you look at the bottom left corner, the chromatin phase, the DNA is very loose here. It's like a bowl of spaghetti. The cell can move this DNA around to find the gene of importance. If you look at the chromosome phase over there, it's going to be very tightly coiled or tightly wrapped up. The phase is used for separating the DNA equally during cell division. This way we can hopefully get two equal sets, one for each new daughter cell that's produced. I think of this as like a newspaper. You want your newspaper. It's easier to deliver them if you still get a newspaper. It's easier to get those when they're coiled up, wrapped up in the rubber band. You can transport a whole lot more of it. Several newspapers instead of just one, because if they're unwrapped or unraveled in the car, then the newspaper person just drives by and rolls down the window and shoves them all out, and it's a mess, and it's hard for you to find and organize what's going on in the newspaper. But if it's tightly coiled, when you're ready to read it, then you can take that rubber band off and uncoil your newspaper and find whatever it is that interests you. Same thing here. DNA is finding that it's transporting the genes of interest so that reproduction can take place in cells or organisms. We look at ribosomes. These are cell particles made of ribosomal RNA or rRNA and proteins. These are not organelles. These are cell structures, but they can still make proteins and enzymes. So these are the site of protein synthesis. They act like the actual construction site for making proteins, not buildings. And normal proteins and enzymes are all made here. There's two types of ribosomes that are going to exist based on location. There's lots of rRNA, or lots of RNA types. So we're looking at these ribosomal RNAs. Talking about two types of ribosomes: free ribosomes and bound ribosomes. So these free ribosomes, they float freely in the cytoplasm of a cell. They're found in all types of cells, prokaryotic and eukaryotic. So these ribosomes make proteins that will stay inside the cell for use by the cell, like enzymes associated with a metabolic or DNA replication. When we talk about bound ribosomes. These are actually attached to the endoplasmic reticulum organelle, or RER. These are only found in eukaryotic cells only, or eukaryotes, because they have this organelle. Remember, prokaryotes do not. So these make proteins that will leave the cell to be used elsewhere. Most are for communication between cells, such as antibodies for fighting infection. So we talk about these endomembrane systems. Remember we talked earlier about endocytosis and exocytosis. So we talk about endomembranes. Endo means inside, system means multiple parts. So let's talk about endomembrane systems. These are pathways found inside of membrane-bound organelles that are involved in making proteins that will be leaving the cell. Membrane-bound organelles also create separate, specialized compartments within the cell to carry out isolated, complex chemical reactions without interference from other components in the cytoplasm. There's lots of folding going on there. So if you think about the ER, or the endoplasmic reticulum, this is a membrane-bound structure. It's composed of a network of small tubes called cisternae. Cisternae just means tubes. They're always found just outside and around the nucleus. And there are two types of ER inside of eukaryotic cells. Smooth ER, or SER, this structure helps with the synthesis of lipids, phospholipids, and steroids. It helps with carbohydrate breakdown, things like glycogen or stored sugar to glucose. It turns it into usable sugar. So it helps to detoxify the blood. Liver cells are located here in the SER. So it also helps to store calcium. It's just needed for muscle concentration. Muscle cells have lots of SER. You've also got the rough endoplasmic reticulum, or the RER. This structure helps with protein synthesis. It provides a safe area for protein folding. Also, ribosomes are bound to the outside of the organelle and deposit the protein inside as it is made by the ribosome. So inside the structure, the proteins can fold up into a specific 3D structure needed for function. If we look at the Golgi apparatus, this is also another membrane-bound structure. This structure modifies proteins by attaching sugars to them. This is what we call glycoproteins. It is kind of like gift wrapping a present to disguise the protein for export through the cell membrane. It's kind of like shipping something to through the mail store. You put it in a box. I'm just going to wrap it up and be able to ship it. These are composed of flattened tubes, also called cisternae. These look like stacks of pancakes. So lysosomes is another one we're going to talk about here. These act like a stomach for the cell, or membrane-bound structure. They are involved in digestion and recycling. That process is called autophagy of molecules. So these are full of digestive enzymes. So if we look at these vacuoles and vesicles, these act kind of like closets for storage of material. Here again, membrane-bound structures. So if we think about phagocytes and pinocytes, they are storage structures for various products needed by the cell. They're going to help to move things into and out of the cell. And you've got various types of these things like food vacuoles, contractile vacuoles, central vacuoles, that are going to help to remove excess water in aquatic single organisms, or at least that's what a contractile vacuole is going to do. You can see the little vacuoles there. They're going to remove that excess water. So look back through this, and we will continue on part 2 of this very soon.